Laboratory Professionals Week. Our next speaker this morning is Sifa. She is presenting on sickle cell.
Good morning, everyone. Thank you for joining me today. My name is Sifa, and welcome to today's talk on red cells ring but sickle sting. So this title encompasses and captures the visual appearance of normal red cells, so the ring-like shape, but it also highlights the painful consequences of the sickle cell shape found in sickle cell disease, which is a crisis, or in this case, it's a sting. So red cells ring but sickle sting. We'll first review the genetic code related to sickle cell disease, then mention the features and epidemiology of the disease. We'll spend some time discussing lab tests and explaining the roles that these tests have in diagnosing and monitoring the disease. We'll talk about managing the disease, and we'll end off the presentation discussing some innovation and some bright future directions related to sickle cell disease. So let's begin with a review on the genetics associated with sickle cell disease. There's a genetic mutation where a single nucleotide substitution within the beta globin gene on chromosome 11 is altered. So the mode of inheritance here is the autosomal recessive pattern, which is important because individuals must inherit 2 copies of the mutated gene, one from each parent, in order to develop the disease. So in this case, 2 abnormal hemoglobin or 2 abnormal genes will both code for an abnormal hemoglobin S. And those who inherit just 1 copy of the mutated gene and the other copy of the normal gene are considered carriers, and they bear the sickle cell trait. The genes code for both the normal and the abnormal hemoglobin, and the abnormal hemoglobin, the hemoglobin S, is usually less than 50%. They're usually asymptomatic, but and they probably don't experience the whole spectrum of symptoms, except in some few cases of high performance sports where individuals with sickle cell have an increased risk about 200 times more of having rhabdomyolysis, which is actually the main cause of sudden death in recruit basic training with sickle cell trait. So sickle cell trait is known to have a heterozygous advantage against malaria, and it protects against severe and complicated anemia. And so these sickled environment is more hostile for the Plasmodium parasite, and also the low oxygen levels doesn't really help either. So the Plasmodium needs red blood cells to multiply, and it's so much more difficult in this kind of sickled environment. So sickle cell disease, however, does not prevent or decrease the risk of malaria, but it actually increases the risk of death from malaria. So the advantage is only with the trait. So here is a diagram that puts it in a picture form, puts the genetic code in a picture form, and you can see that there's a single nucleotide substitution with this gene where glutamic acid is replaced with valine on the 6th position of this gene. And this change causes this change in amino acid causes the hemoglobin molecule to crystallize in low oxygen environment. And this is actually what makes the sickle cell get stuck onto the blood vessels. So unlike the smooth disc-like shape of healthy red cells, sickle cells are more elongated and are crescent-shaped, and this type of red cell is known as a drepanocyte. and the sickle red cells have increased rigidity, which makes them less flexible compared to the healthy counterparts, and this disrupts the smooth flow of blood through the vessels. And because of this abnormal shape, the sickle cells have a decreased lifespan compared to the normal red blood cells, about 10 to 20 days. On a peripheral blood smear, as you can see here, there's a lot going on, but we see the drepanocytes, which are the shape, the sickle shape of the red cells. We see polychromatic red blood cells. We can also spot some target cells, and sometimes you can also see hollow jolly bodies on patients with sickle cell disease. And the polymerization of the hemoglobin S is one of the important characteristics of sickle cell, where, like I mentioned, under low oxygen tension, this molecule polymerizes and it actually forms these long, rigid chains of red blood cells, and it makes them more prone to adhering to the vessels and contributes to vaso-occlusion as well as hemolysis because of this premature destruction of the cells. Both of these consequences is what leads to the crisis, or in this case, sickle sting. So prolonged vaso-occlusion can actually accumulate in the tissue, and when the energy demand is not really matched by the supply, it then goes on to cause inflammation and can cause systemic vasculopathy. Moving on to the epidemiology. In the United States alone, about 100,000 people are affected by sickle cell disease, and it's higher in certain ethnic groups more than others. In 1 in 365 Black or African American births are affected, and also 1 in 16,300 Hispanic American births are affected. And as for the trait, 1 in 13 Black or African American babies are born with the sickle cell trait, which is significant. Globally, Africa bears the greatest burden of this disease because of the geographic link between sickle cell disease and malaria, where there's about 1,000 daily sickle cell disease births. And unfortunately for these children who are born with sickle cell disease, they don't survive until beyond the age of 5. They usually either die from severe infection or severe anemia. And like I mentioned previously, there's this geographic link between sickle cell disease and malaria, but only the trait carriers have a reduced susceptibility to the Plasmodium falciparum. So this gives you an idea of the newborns with sickle cell anemia globally. And we can see that Africa bears the greatest burden of sickle cell disease, where there's over 300,000 sickle cell anemia births just a year. And for the clinical manifestations of sickle cell disease, vaso-occlusion crisis, which I've mentioned previously, is the pain that's usually localized on either joints, sometimes bones, and even the lungs or the chest. And this is caused by the polymerization of the hemoglobin S. Secondly, we have organ damage, which usually happens when there's that prolonged vaso-occlusion throughout the body, and the lungs can get severely affected, which can cause acute chest syndrome. This picture here shows a fat embolization in acute chest syndrome, which is actually the most common cause of hospitalization in patients with sickle cell disease, causing ICU admission. And this fat embolization happens when the bone marrow undergoes necrosis, which is caused by the crisis, and the bone marrow material actually just goes through circulation and it leads to this fat emboli. Other manifestations can include renal dysfunction, nephropathy, rhabdomyolysis, like I mentioned, which can impair the kidney function. Another significant manifestation is hemolysis, which I touched on earlier, is caused by the premature destruction of red blood cells and can contribute to anemia. And we see all the symptoms associated with anemia from that. Again, the trapping of the red cells by the sickled red shape can result to abdominal pain and cause splenic infarction. We'll spend some time discussing the different lab tests as they pertain to sickle cell disease. And historically, the solubility test was a very simple and cost-effective test used to observe the solubility or the insolubility of hemoglobin S and hemoglobin A. And some of the modern techniques used include newborn screening, electrophoresis, HPLC, and PCR. And a version of this solubility test is still used today, and it's used as a screen for the presence of hemoglobin S, where the red cells are added to a sodium hydrosulfide solution, and an agent is used to then lyse, and it releases the hemoglobin. So if hemoglobin S is present, it forms this tactoids, and the solution becomes turbid. So the endpoint of the solubility test is actually in its insolubility, or the insolubility of hemoglobin S. And here you can see the first tube. This is a negative reaction. There's an absence of the sickle hemoglobin, and the tube 2 is turbid, and it shows the presence of the sickle hemoglobin. Electrophoresis is used to separate out the different types of hemoglobin, and in this case, it's used on an alkaline medium, and then it's subjected to an electrical field. Different hemoglobins have different charges, and they'll move at different speeds throughout the gel, and then they'll eventually show up and form bands. So a normal adult, as you can see here, will show up as hemoglobin A primarily, but a very small percentage of hemoglobin A2 as well. Normal newborn, they'll usually show up as fetal hemoglobin with some increasing values of or presence of hemoglobin A. Sickle cell disease primarily shows up as hemoglobin S, and the trait will show up with the S as well as percentages of the hemoglobin A, but with lesser of the hemoglobin S than sickle cell disease. Hemoglobin SC, which is another hemoglobinopathy, will show up as hemoglobin S as well as hemoglobin C bands. So this method is not reliable for hemoglobins that are present in very, very low quantities, like hemoglobin A2, like you can see here, or hemoglobin F, depending. So that's where HPLC actually comes into play. And HPLC is sensitive and specific. It quantifies the different hemoglobin fractions, especially for these low hemoglobin fractions. We can see them, and we're able to quantify them, like hemoglobin F and A2. And it does all this in a single test. So for HPLC, however, some variants of hemoglobin will alter the A1C results. So that's usually a disclaimer that's put out for us when using HPLC. The image on the left here might be a little bit blurry, but it's just to point out the peaks and of these different hemoglobin fractions. And this particular peak shows sickle cell disease with the very high hemoglobin S zone. And to actually accurately quantify this hemoglobin A2 or this hemoglobin F with this extra variant, HPLC would be actually very useful because it doesn't really give us much information about these little peaks. And HPLC again is useful when it comes to individuals who are undergoing transfusion and other types of medication that can affect the different hemoglobin variants that are in small percentages. So this slide shows the HPLC method, which is based on an elution of hemoglobin that's bound to the solid phase over time. So the sample is injected into a column and hemoglobin is eluted, which is like the retention time. And as this elution happens, it's detected, the hemoglobin is detected, and it can detect very different species of the hemoglobin, including the Bart's hemoglobin, hemoglobin E, A2, and so on. So with genetic testing, chorionic villus sampling is done even before amniocentesis is available, which is around 9 to 10 weeks of pregnancy. DNA is extracted and is used to detect mutation through a form of PCR called as restriction fragment length polymorphism. And this method of PCR basically detects the disease based on restriction enzymes. So in this case here, I have this image that shows MS2, which is basically just a type of restriction enzyme that cuts DNA in segments and is separated into number of bands, which indicates the number of mutations. So here for the sickle cell trait, here this is the for the healthy individual, the A. There's 2 bands because there was cleavage, there was cutting that happened. In sickle cell trait, there's no cut made in the abnormal globin gene, so that single band appears. But there's also the normal hemoglobin, a normal gene that cleavage happens, and so we'll see these 2 bands as well as the not cut band. For the sickle cell disease, we see that there was no enzyme cutting that happened because there is that mutation on both of the genes, both of the globin genes, and so this single thick band appears up here. So the lab tests in hematology, as well as the supportive care that blood bank offers, is very important and for individuals with sickle cell disease. And the red cell exchanges and blood transfusions are usually like a lifeline for these individuals because of the crisis and severe anemia. Usually C, E, and Cal negative blood is given to avoid immunization of highly prevalent antigens. And usually an extended profile is done, usually preferably by genotyping, but serology can also be used. So there's this GATA mutation where Duffy B negative red cells are not needed for patients with this mutation because this particular mutation impairs the factor in certain erythroid cells. And so for true null Duffy B patients, they cannot make anti-Duffy B because of this mutation. So they can be given, they don't require Duffy B negative red cells, which I thought was really interesting. So with a constant transfusion, usually steroids or some sort of immunoglobulin is given to prevent acute hemolytic transfusion reactions. And hematology also plays a really important role, which we'll see in CBC, where it can guide or give like a guidance to treatment decisions, as well as assess the disease progression. Here are some hematology parameters in sickle cell disease in comparison to the trait and the normal hemoglobin. And we see that red cells are usually decreased, but in this case it's increased. Usually red cells are decreased in sickle cell disease compared to the trait. Hemoglobin levels are obviously decreased in sickle cell disease due to this chronic hemolysis going on, as well as a shortened lifespan of the cells. Hematocrit usually drops because of this ongoing hemolysis. But we see that MCP usually is normal because it's a normocytic anemia. And white blood cells you can see leukocytosis due to like infection or during this crisis that's going on. And this is just a summary of the table. It's just in a table format. So I thought it was important to highlight the differences and similarities between sickle cell disease and thalassemia, which they share a common clinical manifestation. First of all, they are both autosomal recessive. They both present with anemia. Hemoglobin F is usually increased in both of them. And to distinguish them based on clinical presentation is very difficult. That's why hemoglobin electrophoresis is one of the differentiating factors between the two diseases. So sickle cell disease is a hemoglobinopathy, so it's a qualitative defect. Again, like I mentioned, it's known to be a normocytic anemia. Thalassemia, on the other hand, is a quantitative defect where there's these mutations in the gene that causes reduced globin gene synthesis, but the structure of the gene itself is small, and it's known for being a microcytic anemia. As for the management of sickle cell disease, the therapies either target the genes, so to address the production of the hemoglobin S, or it targets vaso-occlusion, or it can also disrupt the polymerization of the hemoglobin S. So when thinking about ways to disrupt the hemoglobin S polymerization, there's time before, this is time here, as this graph shows, there's time before these polymerized fibers form. And so the goal is to try to get the red cells into the circulation within this time before the fibers form, and then can cause sickling like you see here. And more on the management of sickle cell disease, we have different biomarkers. We mentioned some of the red cell indices, hemoglobin F. There's also CRP that can be used, ESR, interleukins, and LDH as well. Can all be used to monitor sickle cell disease. Regular pain assessments and vaccinations are important too. The vaccinations just help prevent infections, which is important. And the lung function tests, in this case, like the spirometry test, is important because it allows for the detection of acute chest syndrome, which we talked about is the second most common reason why patients with sickle cell disease are admitted to the ICU. So this next slide provides an outline of key therapies in the management of sickle cell disease. I'll just mention a few. For example, this hydroxyurea here works by increasing fetal hemoglobin, which reduces the frequency of these crises. L-glutamine here is an oral medication that is used to help reduce oxidative stress and inflammation that's associated with sickle cell disease. Krisna here is a monoclonal antibody that reduces vaso-occlusion by inhibiting the cell adhesion to the vessels. Folic acid, for example, here is used as a support just to increase the production of red cells. So the future of sickle cell looks very bright. Research on the use of small molecules is underway, which hydroxyurea is the only FDA-approved small molecule for sickle cell trait or sickle cell disease. And point-of-care microchip electrophoresis is important. It is being improved. It has been used in the past, but it's being improved because it helps for the spot analysis and of sickle cell, as well as helps in populations of low resource areas, as well as the pediatric population. So when it comes to CRISPR technology for sickle cell, the stem cells with the mutant gene are extracted from the patient, and through CRISPR-Cas9 editing, it's corrected, and then it's replaced into the patient. And then eventually the cells then engraft and form these healthy red cells. So the two therapies that I wanted to talk about is Casgevy and Lyfgenia, which are the two therapies that are approved by the FDA as of 8th December 2023. And it's approved for patients over the age of 12. And how it works, at least for Casgevy, is that the CRISPR-Cas9 is directed onto cut DNA, and it edits the DNA where it's cut, and it's then transplanted back to the patient, and Casgevy involves a gene knockout. So it switches off the gene that suppresses fetal hemoglobin, and that fetal hemoglobin then replaces the mutated adult hemoglobin. As for Lyfgenia here, the stem cells are genetically modified to produce more hemoglobin A, and then that is returned to the patient. So it's basically a use of like an artificial gene that takes a piece of artificial gene by use of virus, and then using that virus to deliver the healthy hemoglobin A to the patient. So there's also news that CRISPR-Cas12 for the use of sickle cell is going to starting 2025, which basically uses the same technology as the Cas9, but just with different recognitions. And here I just included the difference between the Cas9 and Cas12, like I mentioned, is it uses the same technology where the proteins are able to recognize the DNA sequences stored within this CRISPR patterns, and then it cuts any DNA molecule with that matching sequence. So for the Cas9 uses a different recognizer than the Cas12 does. And in the case of Cas9, the DNA cleavage is blunt. It's like a blunt cut end, while the Cas12 is more of like a staggered cut that's created. And some of the challenges with CRISPR-Cas9, at least, is the cost. Which is priced at about $2 million per patient, which is very expensive. And the treatment is also really challenging to manufacture and deliver. It's promising though, and the use of chemotherapy can be tough on patients, which is needed before administering this treatment on sickle cell patients. It's also very time intensive, which can take about a year or even more. So in summary, today we discussed sickle cell disease, the genetic code, its features, the tests that's used to diagnose and manage sickle cell disease. We also discussed a little bit about the ongoing research, which looks really promising in relation to sickle cell disease. And I hope this presentation gave you a better understanding of sickle cell and helped you appreciate the work that we all do in this field to improve the quality of life for people living with sickle cell. Thank you, and have a happy lab week. And if there's any questions, I can take them.
I don't have a question, but I'm You know, one of the most scientific shows I watched, 60 Minutes, showed somebody who was actually cured of sickle cell through the CRISPR technology. She wanted to be an athlete, whereas she couldn't when she had sickle cell disease. And then afterwards, you know, she was a field and track athlete. So in her high school years. So it is very promising, and I love to see people actually cured of these kinds of devastating diseases.
That's interesting. Okay. If nobody has any questions, thank you for joining us. We don't have any more talks today. Just tomorrow morning is our last talk at 9 a.m. And have a happy lab week. Thank you.
Thank you.